Hello, and welcome to today's webinar held in the lead up to World Malaria Day, which will be marked officially on 25th of April. So we might all be aware that malaria control depends not only on discovering effective drugs, but also on making informed decisions about how and when to use them. And so today's session will, amongst other areas, explore how antimalarial drug discovery and mathematical modeling work together to guide evidence-based malaria control strategies. To take us through this session, it is my pleasure to introduce Dr. Stephanie Kamunya, a malaria researcher with interdisciplinary training spanning antimalarial, drug discovery, and infectious disease modeling. Her work integrates biological knowledge, human behavior, and transmission dynamics to support evidence-based decision-making for malaria control and elimination, with a focus on how drugs, vaccines, and other interventions can be optimally deployed in a real-world setting. Before I welcome her, please note we will have a Q&A session at the end of the main presentation, but also feel free to use the chat to share your questions if you have them. Without further delay, please join me in welcoming Dr. Kamunya.
Hello, everyone. Thank you so much, Annie and your team, for diligently working on this, giving me this platform to talk about what we're going to talk about. So much thanks to your team, and I'm very excited. I'm very excited to give this talk and to just share knowledge with you guys. So as Annie said, my name is Stephanie Kamunya. And yeah, just as she said, I'm trained as a medicinal chemist. We'll get into that. And currently working on infectious disease modeling, specifically malaria. So today, feel welcome, karibu sana. I'm excited. And today we'll go on a journey. We'll go on a journey from the molecules, all the way from molecules, those small tiny things that you heard about in chemistry, some of you back in high school, and all the way to malaria impact. So making sure that these drugs that have been made are used optimally for the most impactful results, right? So at the end of it all, I want us to just understand how drugs and models shape malaria control, right? And because we're in Kenya, we are still in malaria control. And other countries, like for example, Southern Africa, they talk of elimination. But for now, we are talking in a Kenyan context. So because we're getting disease specific, we're talking about malaria. Before I go any further, just because some of us might not be very familiar with malaria, let's just take a few minutes to go through what malaria is exactly biologically. So malaria is the disease, right? That's what we call malaria. But the parasite that causes malaria is what we call plasmodium. Now, there are very many species, but in Africa, and what gives about 96% of cases in Africa is what we call the Plasmodium falciparum, right? You'll get, there's also another specific species that causes malaria in humans, which is what we know as the Plasmodium vivax. But because remember we're in Kenya, we just want to look at the Kenyan context, we'll focus on falciparum, right? Now, this image that you see here in the middle of your screen, pay attention and see that the only way, if you look at step number 1, what introduces malaria to a human being is a bite from an infected mosquito. What does that tell us? It tells us that it is vector-borne. So a human can't give another human malaria, right? You can only get malaria through another infected mosquito. So let's not lose track of that. Now that's why we call it's a vector-borne disease, because it's introduced to the human through a vector. Now, this vector actually are usually female Anopheles mosquitoes, but I don't want to go too much deep into that. But what I want you to focus on is that the parasite is introduced to the human through the bite of an infected mosquito. The parasite then starts in the liver, right? Makes a home there, feels comfortable, divides itself, and then goes and infects the red blood cells. Now that's where we see the clinical symptoms. We see fever, we see, the body, the fatigue, and that's when this parasite is doing a number on the patient's red blood cells. And even that's how malaria is actually detected through, let's say, microscopy. If you take a blood sample, they can see the parasites that are causing havoc on the red blood cells. Now, if you look at step 5, step 6, the parasite then goes on to develop again into now, there's the sexual, but I don't want to go too much deep into that. One thing I want you to take note of is that it's a vector-borne disease and it's a parasitic disease. Why am I drilling this as a vector-borne disease? Because that really affects the interventions that we use in malaria control. Remember, I talked of malaria control, right? How do we control malaria, right? Now, the interventions that we have in place are used to protect that barrier between hosts as the human and the vector. That's why we have things like insecticide mosquito nets. We have things like indirect geospraying. Those are interventions we call vector control, because those ones affect the vector. When you're sleeping in the net, the vector can't access you. When you're spraying your house, the vector can't access you, right? But today, the intervention that I want us to focus on is the treatment. Treatment is just someone who's sick with malaria now gets prescribed a drug. Because, we can't follow the mosquitoes and treat the mosquitoes, right? So it's much more easier, of course, to treat malaria or to treat the human, right? So we have scientists that have worked diligently on this and have produced a number of antimalarials. What you see on your screen there is what is recommended by the WHO, especially specifically the Coartem, that is the artemether-lumefantrine. It's what's recommended by the WHO for treatment of uncomplicated malaria. So that's someone who is, yes, they're infected, but they are not so far gone. But now the other drug you see there, the artesunate, is for treatment of severe malaria. Now that's when the patient is, even you can see it's given in IV, because it's usually given in hospitals, because the patient is bedridden, they can't move. So that's usually the drug. But let me tell you, there are a number of other drugs that are used. But these are the examples that I'll give. One thing that I want us to know, malaria there are around, I think, 600,000 deaths globally from malaria in 1 year alone. And let me remind you, malaria is preventable, malaria is treatable, but still we die. Okay, not we, but still people die. This begs a question. How come? That means there's something behind the scenes that's going on, because the drugs we have are efficient. They're enough. That is someone to go to hospital, they're given medicine. But now we see people still die. What does that mean? It means that sometimes there's something going on behind the scenes that we can't really tell. For example, poor treatment, right? And by poor treatment, I mean someone has gone, first of all, they have sought help in a local chemist. They haven't gone to the health facility, right? That local chemist is going to tell you, by the way, we have this drug you can use, and it won't be a recommended drug. right? So of course, we don't expect complete parasite clearance from you because the drug is not recommended. It is substandard. Another thing actually that usually affects people also is lack of adherence to the drug. You've been told to swallow the drug 3 times a day. Yeah? In 3 days, you've been given a dose to finish in 3 days. But you take 1 day, second day you are better, you leave the third dose. What does that mean? You are breeding chances of resistance, right? So these are also things that affect the interventions that we have today. There's even, just allow me to sidetrack. You're hearing of antimicrobial resistance. Why is that? Because some people don't adhere to the drug. So it's not only in, microbial means general parasite, but also we also have drug resistance in malaria, right? So now that we have seen that how treatment works, have you ever thought, wow, these drugs, they are good? Have you ever thought of the work that might have been taken to undertake to put those drugs on the shelf? Now this is what that I want us to dive deep into. Most people, I'm sure, on this call, most, if not all, know of modeling. And as Annie said, yes, I am an infectious disease modeler. And most people are familiar with infectious disease modeling. But today, I am so excited to introduce you to molecular modeling. So just give me your attention just for a few minutes as I try and guide you through this new found concept of molecular modeling. And I'm excited to do that because that's what I was trained on. So before even we do that, some of you might be asking, wait, molecular modeling, what is a molecule, right? So some of us, of course, at one point in our life, we've gotten sick, right? We've gone, some of us go to chemist, some of us go to the hospital and we are given a prescription. We go and fill it in the chemist, right? Go to a pharmacy, right? And yes, you open the drug, you are so sick, you just want to pop that tablet. But do you know that there's a paper inside that box? Have you ever bothered to open it and just check what is it saying, right? Now that next time, maybe when you open, you'll remember what I'm saying. Now that paper is usually what is commonly referred to as a patient information leaflet. If you open that paper, now sometimes you will find a nice chemical structure drawn there, as you can see on the diagram on your right hand side. You can see a nice chemical compound drawn there, and seen. And so actually, that's what the drug is. That's usually the chemical structure of the drug, right? That is what we call a molecule. Okay, I hope now we're on the same page. Usually, a drug, the chemical structure is drawn, and that's what we call a molecule, right? Now, molecular modeling, just as infectious disease modeling is mimicking the movements of an infectious disease across a population, right? We are mimicking the movement of an infectious disease when you do infectious disease modeling. Molecular modeling, yes, you get it, mimics the movement of a molecule. Now we are no longer looking at population level. We are going down deep. We are going now to the atomic, not atomic, because atomic is the Cl, those things. But we are going deep down to those minute levels, right? That's what we call molecular modeling. Now, I've shown you what a model is, what a molecular is, what a molecule is, right? Now, that structure that you have seen there, how do you think those experts came up with that structure? Did they just draw it once and say, yes, we found the solution. This is what we are going to use. That's not what happens. It's very taxing. Traditionally, they would draw up to 1,000, and not even draw, synthesize in the lab. Very hard task. They would synthesize all those molecules, test them against the disease, the parasite, virus, bacteria, and check the potency of each compound. Now, by potency, I mean how it kills the compound, right? So some will be more potent than others, and that's how a compound is progressed through. That's how we eventually get to the drug that you have in your homes, right? But now, let me tell you, biologists are doing their things. Computational scientists are doing their things. Technology is working. Technology is booming. Why do I say that? We are able to take the parasites of malaria, put it under those giant things, what biologists do, identify targets that are important for the life or for the sustainability of the parasite, right? Now, let me just say that again. For me to put my finger up like this, or for me to put even my finger through my nose, what does that mean? A signal has been passed from my brain ahead to the bones, the nerves. The nerves are now saying, put your finger to the nose, right? There's a signal that has gone through. For that parasite to survive, there's a signal that those proteins inside the parasite talk. The DNA, the things that make the parasite. There's a signal that makes sure the parasite survives. Even for you, while you are breathing now, the breath you're taking in now, there's a signal that the brain is sending to your lungs to draw breath. It's the same thing for a parasite. Now, because of the advancements of technology, let me tell you, these biologists are able to, with their omics, they're able to take apart a parasite and say, aha, that protein is important for a certain signal for the parasite to survive, right? So nowadays, target identification. Those are studies that identify targets that are very crucial to the survival of the parasite. What does that mean? If we hit that target and we strike it down, the parasite does what? It dies, which is what we want to do with malaria, right? So that is what molecular modeling is. It now takes advantage of that, because now we are able to draw the molecules in 2D. Now we have computational chemists who are doing it in 3D. In 4D, in fact, we'll just see. But the main thing that I want you to get from this, we are able to identify targets. From that target, as you can see here in this box here, let me try and put a pointer so that you can all see. In this box here, what we are calling a molecular target, right? You see in this box here, it's just looking like a big blob, right? But in those target identification studies, they identify a pocket, what we call a ligand receptor complex. They identify a pocket, which is very important for the survival of that target. And once we design a drug that fits perfectly into that pocket, what happens there? The target dies and the parasite dies, which is what we want our drug to do. And that's how molecular modeling comes into place, right? We are able to fast track and see, does this compound really fit that pocket? Does this compound really target that? Does it really fit well? We are able to do that in our computers, what we call in silico in our computers. And that is molecular modeling, right? So you can do it through molecular docking or dynamics. Now, so that's the same thing, Docking is a picture, dynamics is a video. We will actually see it, what I mean by molecular dynamics. But molecular dynamics, I tell you, is computationally heavy. It's very heavy. So, but docking, as you see, it's rigid and flexible. It's just a picture, and we just see, okay, the drug fits in the pocket. Wow, good. Now, it's all good in theoretical. Wow, like I can see you have made a chemical compound, a molecule. I can see it fits perfectly. Wow, but you can't take that to anyone. So what does that mean? We need to synthesize that compound and have experimental evaluation, right? Why molecular modeling is very good and why I really enjoy it is because, remember, traditionally we were doing it blind. We're just saying, oh, let's move this Cl atom and put it here, let's move it here, let's put it here, and let's test. But with molecular modeling, we are able to take advantage of that. Instead of synthesizing 10,000 and having a positivity rate, that is active compounds of, let's say, from 10,000, you only get like maybe 10. Molecular modeling helps us increase the positivity rate. We can have from maybe 10,000, we can get now, let's say 500, right? And that's the flashlight that we need in the dark. So that's what molecular modeling is. So as I mentioned, there's molecular dynamics. And that's also to take you back. Molecular modeling mimics the movement of the molecule, which is what we will see here. Because remember, nature is ever moving. It's not stagnant. It's not just here. So that's how molecular modeling, one of the things I mentioned was molecular dynamics. And from this video, you can see what I mean by molecular dynamics. You can see that molecule moving, accessing the pocket. You can see how it negotiates with the target. You can see, right? Yes, you can see. So molecular modeling gives insights. Is synthesizing this molecule worth it? As you can see, this molecule here is accessing the pocket very well. It's negotiating with the target, and it's able to access the pocket. Wow, look at it going through. And it's actually sitting very well in the pocket, right? Remember, molecular modeling mimics the movements of a molecule, and that's what we do here. And that's what we are doing here. Allow me to, yes. As Annie mentioned, that's what I did. I did this I did this previously, as Annie mentioned. And it was very interesting. It's very interdisciplinary. Remember, I mentioned the biologists who do very good work. I mentioned the medicinal chemists who are in the lab working, synthesizing, right? So I'll just take you through what we did in this paper, right? We'll start here. What we did, there was an anti-cancer drug which is the parent compound here, this molecule that I'm circling here. There was a parent compound, and it's actually currently an anti-cancer. This anti-cancer was tested for antimalarial activity and was found to be active. It was done for target identification studies, and by that I mean it was exposed to targets, and we were able to deduce which targets it inhibits in the malaria parasite, right? So we found out that it actually inhibits what we focused on in this study we focused on two targets, and let me take you back there, which are structurally kinases. I won't dive deep into that. Just know that our targets are kinases, right? Now, we focused on two kinases. The phosphatidylinositol 4 kinase, beta. Don't focus too much on the names. Don't worry. You can just let them pass you by. But just know that we've had two kinases, two kinase targets. The phosphatidylinositol 4 kinase and the cyclic GMP dependent potent kinase, right? Those are the two targets. Keep that in mind, two targets that we were targeting, okay? And also keep that in mind that you are dealing with an anti-cancer. Remember, anti-cancers just target the human things, right? Remember, I told you, molecular modeling helps us design the molecules that we need to synthesize, right? So for once we had this compound, we were like, okay, wow, let us sit down and design other compounds. So what we started with, because you also need to be systematic. You can't just change everything on the parent compound. You need to be systematic. And also, do something that's chemically accessible. You can't come here and change this NH2 and my friend. It's not stable in the lab. So do something that's chemically accessible, right? What we call synthetically accessible. And it was actually this method that we see here, which is actually what we changed to an aromatic compound. Don't worry. If you are not understanding those chemical compounds, don't worry. Just let it pass you by. But just what I want you to focus on is that we were able to design the compounds, right? We have our compound library. We had very many, actually around 500. Then, remember I told you, we are now doing a structure-based drug discovery. We have our target, actually not structure, but target-based drug discovery, right? So what we have here, what you can remember, we have two targets. What we have here in green is our target A. What we have here is target B. I don't want to name because I don't want to you guys to disappear. I want to keep you captive, right? I want to captivate my audience. So we have target A and target B. So remember, molecular modeling helps us identify compounds with a very good structural pose. It's not viable to do molecular dynamics for all, let's say 500 compounds. It's not available. So the first step that we do, we dock them. So we have these compounds, we design them in the library, then we have the structure of the target, we now throw those molecules in there, we dock them. We try and see, does this one fit, does this one not fit? Is this one touching, this one not touching, right? So that's one thing that this helped us be more targeted and not design all the 500, and not synthesize all the 500, right? So we filtered out compounds with the best pose. Because there were some, like for example, let me look at my cursor where it's going. There are some where this aromatic ring was going upwards, flying up. I mean, which is not possible. But bending like this is very possible because you can see it's trying to access this amino acid and form that hydrogen bond. Now, molecular modeling will have hydrogen bonds. Are you seeing these broken yellow lines? These broken yellow lines that you can see here, we love that because it shows that it won't move from there. Once you bond, it's difficult to breach. Now keep that in mind, right? Now with that, this is the step that we did. And remember, I told you, you can't just take things and say, okay, this one had a good pose, and you're given a tip. We need experimental validation, right? So we took our best chemicals, the compounds, the best pose. Then we synthesize them, which you can see here. So this is just an example. If you want to go deep, you can go to the paper at your own time. But this is just an example of the results that we got. Remember, we are screening for potency. Potency usually means the lowest number here, the better, because it means you don't synthesize kgs of a drug. They say small. So when we do drug discovery studies, the smaller number here, the better, the more potent, the more active. So let me just take you just a little bit, a step just closer to what we were screening for, what we mean by the results, right? Now this diagram, I just want to tell you what we are playing around with, what I meant by just replacing the aromatic rings. But yeah, this is what I meant by that. Now, drug discovery is you need to be a multitasker. From this one compound, you need it to be an antimalarial. You need it to be selective. You need it to be safe. You need to be also available. Because imagine if you swallow a drug and all of it is dissolved in your stomach. That means there's nothing for the blood to carry to go and to take the drug to, right? So there are so many things we balance when we do drug discovery. Now, for this project, what we did, remember we're dealing with kinase inhibitors. Now, you know, as human beings, we have kinases. The parasite also has kinases. Structurally, they are almost like the same. What does that mean? We have one more caveat now to also look for when we design these drugs. We have to make sure that this drug is selective only to the plasmodium kinases. We don't want it to touch the human kinases. And remember, we're dealing with an anti-cancer. Anti-cancer is just there to tackle human things. So we had to juggle that. So we made sure to screen against the human kinases and also the plasmodium kinases. We also screened because this our parent compound was also it's called mTOR, mammalian target of rapamycin. It was an mTOR inhibitor. We also need to make sure that the compounds we made, we need them to be selective only for the plasmodium kinases. We need it not to be active against the mTOR, right? What I want you to take home from this slide is drug discovery, you need to juggle very many things. Number one, activity. Number two, safety. Number three, bioefficacy. What I mentioned. You want your drug, or if you can see here, we also did in vivo proof of concept studies. What does that mean? We used a mouse model. We exposed that mouse to the drug and made sure that at least the compound that we give is also in circulation. Imagine if you have a drug, potent, tick, everything is good, but when you expose it, it's not bioavailable. You only get 20%. That means your body's only left to 20% of drug in circulation, which we don't want. So usually that drug is screened out, right? So please remember, drug discovery, we need to juggle very many things, right? Now, drug discovery is a very long process. What we have just gone through is just the discovery phase only. It's a very long pipeline. From the discovery phase, we go to the preclinical phase. This is where you find now, as you can see here now, this popping, which is now where we expose what we call the lead compound to animal models, making sure that the results that we were getting in vitro, that is when we are outside of the path of the human, right? In vitro outside, in vivo inside, right? So inside we test things like bioefficacy, that's making sure that it's bioavailable. In vitro means we've already extracted the cell and everything. We are just now trying to see its activity. It doesn't have to go through solubility, doesn't have to go through so many things. Now, preclinical makes sure that what we have seen in the discovery phase is also applies to the preclinical phase. Now, clinical trials, I'm sure most of us are familiar with them, having gone through the COVID scenario. Clinical trials usually are 3 phases. Now, phase 1, we expose the drug to healthy individuals. This is just to make sure that our drug is not toxic. This is mainly a safety check. We just want to make sure, okay, this drug is not giving any adverse effects to healthy people. Once we are tick on that, we move on. We now go to phase 2. Now, phase 2, we introduce the drug to the sick people. So if it's malaria, if it's an anti-malaria to people, it's malaria, and so on and so forth. Phase 3, the only difference between phase 3 is we're also dealing with sick people, but now the population increases. In phase 2, if we had only 100 people, phase 3 will have like 100,000. Just to make sure that we still see the results when the population is bigger. Now, let me tell you, to get to that phase 3, just say hallelujah, amen. It's difficult. It's difficult. So to get to the regulatory part in the post-market, that one is a breeze. If you've gone through up until phase 3, don't worry about the rest. What I want you to take into consideration is the hard work that goes into making these drugs that you see on your shelf, in your house, in a pharmacy. If you look under there and add up that time scale, it can give you decades. The work that we've just gone through with the parent compound and such took around, let's say, 5 years, right? That's just discovery, leave alone preclinical, leave alone all those other things. And also another factor is money. Some drugs are good, but they don't have the money to progress. So that's another factor. One thing in our malaria toolbox that went through all that phases was what we now have as what we have now called and have in our toolbox is the malaria vaccine. Now let me just pause and tell you that it took us 40 years. It took over 40 years for it to be where it is now. For those of you who are 40 and younger, let us just appreciate that journey that it took. That's also malaria, or rather, drug discovery is a very taxing thing. So let us just appreciate to have the malaria vaccine with us, right? Now, the malaria vaccine is quite different as to other vaccines, as that it does not provide complete immunity, only partial. So that's something to take note of. Another thing, remember I said it's a tool in our toolbox. It's not the only tool. Now, malaria is complex. If I take back to the first, remember we're dealing with mosquitoes. When you're sleeping at night and you hear that sound in your ear, you hear a nuisance and you do like this. And switch on your torch and you look for the mosquito. Where are you? You won't find it because now it has gone. Now, try and deal with that now with a vector-borne disease. It's very difficult, right? So let us just say that these tools, they need to work together, right? As I've said, it took us 40 years just to have the malaria vaccine in that tube. Now, drug discovery is well and good, but remember it's in a controlled environment, right? So it's going to give all those ticks. But what happens when you expose it to the real world? There are very many things that the decision makers have to take into consideration. For example, access to the drug, the behavior of the people. Like, will they even want the vaccine? Will they want it? Okay. Will we give it to everyone in Kenya? So there are certain questions that need to be answered. And remember, it took us 40 years just to have this vaccine. Don't you think we need to think a bit more and making sure that we are careful in how we implement this intervention? So we need to make sure that we cover all steps, right? It took us 40 years to have this vaccine. We can't just deal with it like nothing. We need to make sure that we give it optimally and that it's impactful. So our decisions need to be evidence-based, right? And this is where malaria modeling comes into place. Remember I mentioned, first of all, remember molecular modeling mimics the movement of a molecule. Malaria modeling mimics the movement of malaria disease in a population. Please, let us remember that. Let us, if you forget anything, let's remember that, right? Now, malaria modeling. With that, we are able to take into account the vector behavior. We're able to take into account the behavior of the human beings. Remember, because not everyone who gets sick will go to the hospital. We're able to take into account how the disease is transmitted. So instead of setting up everything and saying, oh, let's go to a clinical trial of how many people go, of those what if scenarios that we can easily, so that are very expensive to run, right? We can easily check what if scenarios in the comfort of our laptop by just setting up a malaria model, right? Can you see the advantages there? Remember, I told you we have a vaccine that took us 40 years. We also have the real world. So how best can we make this vaccine impactful? How best can we use it? Don't you think malaria modeling will lead us to that answer? I definitely think so. At least it will lead us in the right direction, right? So, and that is what we are doing here at CEMA. Here at CEMA, we have developed a model that has taken into account the effectiveness of the vaccine. Currently in Kenya, we have the RTSS vaccine. So we've been able to create a model that takes into account all that human behavior I've told you, adherence, access to care, all those things that, on the ground that are happening that we don't see in clinical trials. One thing that I want us to also mention as we try and integrate these two, molecular modeling and malaria modeling, is that laboratory efficacy is not always equal to population impact, right? We've been told this vaccine is 100% effective, but on the ground, we only think it's reducing by 20%. Why is that? Malaria modeling will help us answer that question. So that's what we're doing here at CEMA. And just to give you some insights of what our model involves. What it usually does, our malaria model, of course, I'm sure I'm now speaking the language of most people. It divides the population into different groups: susceptible, exposed, infected, asymptomatic, recovered. We use the epidemiology of the malaria disease. And in that model, we're able to take into account seasonality. Malaria is seasonal. Remember, I told you mosquito plays an important factor. So we take into account seasonality. We take into account now the demographics of the people, right? And this model has been fitted to data. So this is what we are using to just make sure that the malaria vaccine that we have rolled out in Kenya is impactful. Man, it took us 40 years to have it. Let us use it well, right? So in summary, drugs alone do not work. Remember, laboratory efficacy is not equal to population impact. Modeling, of course, both molecular and infectious, helps us cut time, right? Remember, before they used to synthesize like almost 1 million just to get one compound, one drug. Right now, we're able to be more targeted, right? And integration is essential for impact, right? We have a vaccine that took us 40 years just for us to have it. That's why Stephen talk about it. 40 years. Let us make sure that we are squeezing out what we can get from it. So thank you so much for being my audience. Thank you so much for just attending. And asante sana. Thank you.
Amazing. Thank you, Dr. Stephanie, for that animated seminar. The talk was amazing. And guys, we're in our Q&A session. Please unmute to ask a question, or raise your hand to pose them. And Stephanie, some of the comments are, she's such a great teacher, which I agree to. Yeah. Guys, now is the time to ask your questions. And maybe as we wait for guys to ask their questions, Stephanie, I just wanted to post one for you in terms of like trust, especially from the population. When you're introducing like a highly effective malaria drug or vaccine into a community, and it's a low trust community, and you've taken us through like the drug discovery, the process is quite long, as you've insisted and made very clear. So I'm just wondering, when you're doing malaria modeling, do you take into account such scenarios, especially on trust, now that we are getting a lot of hesitancy of vaccines? Or how can our health program overcome such?
That's a wonderful, wonderful question, and usually a very common problem. So remember, when it comes to approaching a population, we can't just go there with the white lab coats and say, guys, by the way, spend this much of time in the lab, take this drug now. We usually what they usually do is they invest in community engagement. So they usually recruit people from that population, people who they're familiar with, train them on this medicine, and then encourage them to now go forth and talk of the drug, right? So usually in those scenarios, we take advantage of community engagement, making sure that on the ground people know what is going on. So it's an interdisciplinary thing, and that's where we will use our social scientists, right? We need to work with them. This journey is interdisciplinary in terms of any form of disease elimination. You need to use. You can't just be scientists and say we can do this alone. You need to engage social scientists and people on the ground who are actually doing those things and who actually talk to the population. So yeah, in a nutshell, that's what happens. Yeah. Thank you, Annie, for that.
Paul Corelli is asking, could you please speak a bit about the computational requirements for molecular modeling?
Hey, that's a very good question. That's a very good question, Paul. Now, usually in computational studies, computational drug discovery, for what we call the LMICs, it's unreachable. Because usually that, like I can tell you, the software that I was using costed tens and thousands of dollars. So, and just to run that software, you can't run it on a local computer. You need. I was using a Linux machine that was housed there in the campus. So it's very heavy, leave alone the capacity of the local computer. The software itself are very expensive. We haven't talked of molecular dynamics. Molecular dynamics, I'll give you an example. My simulation, I would run it for maybe 200 nanoseconds or something. Days. But that was before I was exposed to the HPC in Cape Town. But by then, I had moved progress with the work, and I was synthesizing my compounds, the ones that I designed, and I was doing the assays. So that was like later on. And then also, how will you attach a molecular dynamics file to a thesis? So yeah, that's just an insight to what it entails. So the software itself is very tricky and very expensive. Yes, Paul.
Just a follow-up to that question. You mentioned that the software is expensive, but then are there any open source free software tools available?
I like your question. Thank you for that question, Paul. So currently I'm doing a drug discovery project, and now this is now me, just like now myself, right, outside of the access to the software. And most open source tools for molecular docking are not there. But I think you can access, they can give you a special license, academic something, but for molecular docking, I don't have. But for like physical chemical properties, so by that I mean let's say number of hydrogen atoms, those physical chemical things that you need to do with a compound. Now that one I use AI open source tools. Now let me talk of a very good thing that has happened recently. We have what we call Medicine for Malaria Venture, MMV. They have designed a very good tool that's open source. It's called DD4GH, Drug Discovery for Global Health, that's targeted to LMICs such as us. And actually, I was granted a license just the other day. So that's one of the open source tools that are there. So, but in terms of molecular docking, where I can get a target, make that target, 3D, like what we were doing before. Because usually you find it on the web, just amino acid sequence. So the software will take that amino acid sequence, 3D it into those folds that you are seeing, and then now run the simulations, the docking simulations, that is put the compounds in the target. There's no open source tool for that. You need a license, and we're using Schrodinger for that. Yeah.
We have
Thank you.
another
Thank
one
you for that, Paul.
Awesome. Great. We have another one from Bongajun, Ginny Victor. With the reported CULT13 gene mutations in Plasmodium falciparum linked to artemisinin resistance from the greater Mekong subregion and East Africa, what are the new drug molecules already on the way to tackle this concerning antimalarial drug resistance?
That's a good question, a very good question, because drug resistance is something that's really plaguing our current drug tools, what we have in our disposal. So I'm not privy to the drugs that are there. So allow me to go back and do research on that. But I know MMV are doing, that is Medicines for Malaria Venture. They've become very open source. You can already tell the drugs that are in the pipeline, that are lead optimization and all those things. Now the mechanism of action, and by mechanism of action I mean like what is mentioned, how a drug works, right? Some work the way it's mentioned with the CULT13 mutation. Some work with that. So some of that information may not be there in terms of a drug that is still in development because of the patent issues and stuff. So I am not privy to that information in terms of the drugs that are in that pipeline with specifically that mutation. Allow me to go back and see, but I don't have any information that are in the pipeline, those drugs. Yeah.
Okay. And Motinda is asking, I was still in tune to see the model itself. Does it take the compartmental approach of infectious disease modeling? Is the question clear? Or she needs?
Yeah. Good question. I like the enthusiasm. And just as you said, stay tuned. We'll definitely give a talk on the model. And yes, it's a compartmental model. It's not an individual based. So just as she's asked, we're dividing the population into compartments, just as I mentioned before. So so it's a compartmental model. And yes, stay tuned for sure. We'll present on that soon.
Still work in progress, right? Or almost,
yes.
right?
Yeah.
Okay.
Yeah.
And Daniel Njenga is asking, is molecular docking same as virtual screening?
Hi, my learned people, you guys are doing good. Yes, it is. Yes, it's virtual screening. Yeah, is the one. Yeah. So virtual screening definitely is a tool. For example, people who are exposed to millions of, databases with millions of compounds, and they have a target. So they can take those millions of compounds, that database, and just screen it to the target, the 3D target that you have, that pocket that you have, and virtually screen out, virtually filter out the compounds that are of interest. So yes, and I'm very impressed with that question. Thank you.
Amazing. Ahmed.
Hmm.
Ahmed. Okay, that's a personal question. I think I'll attend to that later. So thank you everybody. If there are no further questions, I just want to appreciate Dr. Stephanie for that session. I'm smiling because it was so animated. I learned so much and I was so entertained at the same time. So I just want to thank you so much for making time. And I think the modeling work that is still work in progress, maybe we'll make time once it's out for her to present on that, or for the team, because it's a teamwork to present that. But I really appreciate you for taking us through what it takes to make the drugs, from molecular modeling to the testing, the whatnot. 40 years, I mean, quite a process for drugs to be made. And thank you all for attending. Thank you, Dr. Kamunya, for the session. Until next time, have a wonderful rest of the week. Bye.